Okay, class, so now we're on to part B, and we'll start off by talking about some of the drugs that actually affect actin and microtubule dynamics. And here's just a little table showing some of them. And one of the ones I really want to highlight here is just Taxol, because Taxol is actually um, a great chemotherapy uh, drug to basically treat cancer. And what it does is it binds and stabilizes microtubules and actually can prevent cancer cells from going through the cell cycle. And here's the um, chemical structure of Taxol. The brand name for the chemotherapy is called Paclitaxel, and I believe it comes from the yew tree and is just isolated. And what it does is it binds to the microtubules and stabilizes them. Okay, so here's just a typical cell, and then if you add Taxol to it, it actually creates a really stabilized microtubule formation in them. Okay, so just a cool little tidbit for you guys. But these are actually used in the laboratory a lot to study um, microtubule and actin dynamics, the ones that are specific to actin. Um, so onto nucleation elements, okay? And I remember I mentioned that both actin filaments and microtubules have proteins that help the nucleation event. Okay, and in the case with microtubules, it's this molecule called gamma tubulin. Okay, and it's shown here in blue. Here's actually an EM of the gamma tubulin, and it's basically involved in sort of nucleating the microtubules, and it allows them to just grow on their plus end, and there's no growth or shrinkage on the minus end. And this becomes really important in certain processes in like the cell cycle, and just in general, the microtubules are actually all connected to the centrosome within the cell, and the centrosome contains two pairs of centrioles, and then there's a whole bunch of gamma tubulin around this, and this is what's called the microtubule organizing center. Okay, this is where all of the microtubules are organized from, and they shoot off from the centrosome outward in the plus direction like this. And this is basically what's sort of considered a mitotic aster, where you have your centrosome and then the microtubules sort of being nucleated by gamma tubulin and growing off the plus end. Um, Here's just an image of a mitotic aster from C. elegans, and here you can see the gamma tubulin in red, and here you can see your centrioles that make up the centrosome in the cell. If you want to see a nice EM of the centrioles, here's what they look like. And they're basically made up of gamma tubulin as well, and other proteins. And when we cover the cell cycle, it's not really understood how these sort of replicate and divide, but it's sort of an interesting question that remains in science. But here's just a really sort of pretty EM of the centrioles. And if you want to sort of see what they look like, they're these basically tubule structures that look like this, and they're all made up of tubulin. And so it turns out that if you actually take a mitotic aster and you put it in sort of a gel matrix, it's always going to migrate to the center, right? That's how it got its name, the centrosome. So here you can sort of see a time-lapse video of the centrosome, and eventually it's going to actually move into the center of this matrix here, because the microtubules will grow in sort of equal lengths throughout and sort of attach it to the end. And so here's just a centered microtubule organizing center, or a mitotic aster as it's shown here. And once again, the microtubules are all relatively the same length coming off of the centrosome. Now it turns out though that you don't actually need a centrosome to have a microtubule organizing center. They can form on their own without the the centrioles or the centrosome. So if you have basically a cell and you sort of cut it across, you've got your sort of severed cell fragment, and it turns out that the microtubule organizing center without any centrioles will actually form here and be sort of centered in this, just this fraction of the cell that's there. Now, just like with nucleating factors with the microtubules like gamma tubulin, actin also has nucleating agents, and they're called ARPs, for actin-related proteins. And here's basically the structure of actin here, and here are two actin-related proteins involved in nucleation of actin filaments, ARP2 and ARP3. You can see that structurally they're very similar, and these are involved in nucleating the actin filaments. And oftentimes the nucleation sort of occurs at the sort of the cell cortex. That's the region just below the plasma membrane. And so we'll see in a few slides coming up, as well as next week's slide, how these nucleation under the plasma membrane actually help with lamellipodia formation and the actual cell movement. But here's just a little schematic about how the nucleation of the ARP complex works. Here you'll have your inactive ARP complex with ARP2 and ARP3, then you'll have an activating factor come in, and then that brings them together and serves as a site for the actin monomers to associate, and so you end up getting plus end growth, and there's no loss at the minus end. And so ultimately the ARPs are just basically actin nucleating proteins, and it allows growth on the plus end of the thing. And they function in such a way that they sort of cause these branching, and this creates like a gel-like matrix in the right sort of beneath the plasma membrane, which allows the lamellipodia and other actin-related structures to form. And here you can see the ARP complexes. They sort of come off at a 70-degree angle and form this sort of gel-like matrix on them, which then, as they grow, it tends to push the cell forward. And here's just an EM of the branch points up top here, and here you can sort of see the cryo-EM structure where you have basically all of the actin and then the ARP complex coming off of it, and then a new actin filament coming off of that. And it creates these branch points, which once again got about 70 degrees of angle here. So if you have, here's just a nice image of actin in plants, okay? And so it helps sort of sew the plant cell together, okay? And if you have a mutation in the ARP complex, the cells are actually a lot smoother, okay? So they don't have any nucleation because you're missing the ARP complex in these plant cells. Another protein involved in the nucleation are these proteins called formins, okay? And they actually help the growth in the plus end, and you can almost sort of think of it as a ratchet as it's sort of moving up. And it's a dimer, and basically it moves and adds actin subunits as it moves along. So there's 15 different formins in mammals, and they associate with the plus end and really sort of help it grow at the plasma membrane, and they're sort of indirectly associated with the plasma membrane. Another set of proteins that helps the growth is this protein called profilin. And so in non-muscle cells, basically about 50% of the actin is a free subunit, and then 50% is in an actin filament. And the high concentration is actually maintained in the cytosol by this protein called thymosin. And if you want the filament to grow, the thymosin is actually replaced by profilin here, and then the profilin can interact with the formins to help add the actin onto the plus end of a growing filament. And here's the mechanism by which that works. Okay, so you have your formins on your actin filament, and then you have these formin whiskers coming off that are associated with profilin, and profilin is binding the actin, and so what it does is it basically stacks them on as the foramen is moving up, it will stack actin filaments on. And that's really important for growth of the lamellipodia. And we'll see that. And it's repeated over and over again so that you get growth and sort of movement in it. Now, in addition to sort of the nucleating agents, you also have severing proteins. And catannin is a microtubule severing protein. So if you look in this slide right here, you have Taxol stabilized microtubules. So these are microtubules which are mixed with Taxol, so they're quite stable. And then if you add in some of this catannin protein along with some ATP, it actually severs them in the middle. So it sort of breaks them. You can almost think of it almost like a scissors that's cutting up the microtubules. And there's other ones too that are also sort of regulated by calcium. There's the gelsolins, and if you release calcium or high levels of calcium, they can actually sever the microtubules as well. Now, in addition to the nucleating proteins and the severing proteins, there's also microtubule associated proteins, and these MAPs can actually help stabilize the microtubules. And a really sort of classic example of a microtubule associated protein is this protein called tau, and tau forms the tangles in Alzheimer's disease. So you have your plaques and tangles in Alzheimer's disease, and tau actually forms the tangles. And if you look here, tau is shown in green here, and just MAP2, which is a different microtubule associated protein, is actually shown in orange. And here's sort of a schematic of sort of what they do. So MAP2 actually can help cross-link two microtubule filaments together, so they create this sort of ordered array. And the same thing with tau, except in the case with tau, it's smaller and the microtubules are brought closer together. And typically these are regulated by phosphorylation to help sort of organize all of the microtubules in the cell. Same thing sort of happens with actin, where you have proteins that help to stabilize actin as well as help to sever it. Okay, so tropomyosin binds to the actin filament and actually stabilizes it, where you have cofulin, which tends to destabilize it. And the way that cofulin works, it sort of creates a twist and it sort of snaps it apart. Okay. And in addition to the nucleating agents, the severing proteins, and the sort of associated proteins, there's also capping proteins. Okay, and so what the capping proteins do is they help prevent shrinkage on the plus end. So you'll have the nucleating agents on the minus end and the capping agents on the plus end, and if you just have the capping agents, you're only going to get shrinkage on the minus end. So the microtubule will only shrink on the minus end. So if it's nucleated down here and it's capped on one end, it actually won't change at all. But you see, you can sort of see here that you know, when you have the critical concentrations where you're not getting any growth, the one when you have the capping protein actually changes in length much slower, right? It can't add to the plus end, and it can only sort of shrink from the minus end, versus when you have an uncapped one, the population can sort of grow much faster out, because it can grow on both the plus and the minus end from it. So, so one important capping protein is CAPZ, and when we look at motor proteins in the muscle, you'll see that this is a very abundant protein in the sarcomere, because it actually binds to the plus end of actin in the muscle Z band. Now, if you go back to part A of today's lecture, and we talked about dynamic instability, well, there's actually proteins that sort of facilitate the dynamic instability, okay, and cause destabilization. These are called catastrophe factors. All right, so we've seen MAPs where they can bind to the microtubules and keep them sort of stable, and so they're a lot less dynamic, and so you don't really have catastrophe events. But you can also have catastrophe factors like this motor protein, kinesin 13, which can actually help pull the microtubules apart and increase the rate of dynamic instability. And kinesin 13 comes into play during the cell cycle when the microtubules are sort of out searching for the centrosome or the centromere on chromosomes, okay, because it wants to find those. And so basically the catastrophe factor, kinesin 13, helps increase catastrophe events in the cell. There's also plus N tracking proteins, or called plus tips. Okay, and these sort of bind to the plus end of a growing microtubule. And these can help modulate the growth and the shrinkage of the microtubules. So if you look at this cell right here, you can see the plus end-directed tips are sort of shown in blue and purple here. There's actually, they're stained in blue and red, and so some of them look purple due to the combination of both. In Schizosaccharomyces pombe, which is a yeast cell, it's a fission yeast, you have a plus tip protein called EB1, and you can see it here, it's shown in red, and it just binds to the plus end of the microtubules. Okay. You also have intermediate filaments, and it turns out there's cross-linking proteins with the intermediate filaments that help cross-link the intermediate filaments to other cytoskeletal proteins. Okay, so this protein plectin can cross-link the intermediate filaments to the microtubules, and that's shown here in green. So here in blue you have this intermediate filament, and then in red you have your microtubules, and plectin here is actually cross-linking the intermediate filaments to the microtubules, and that helps give the cell a lot more rigidity. Okay, so now we're going to just move on to the organization of actin because it's actually highly organized, and depending on where the actin is in the cell, it will do different things. So if you look at this sort of schematic here, you have actin organized in sort of a stress fiber, and then sort of right underneath the cell cortex here, you have sort of a gel-like network where you have the ARPs sort of forming, and formin and profilin adding the actin subunits, and the sort of lamellipodia forming. And then you have these tight packed bundles that form filopodium. Okay, and so the organization of actin is done and controlled by these actin cross-linking proteins, and here's just some examples of them. There's spectrin here, it's actually a tetramer, and then you have fimbrin, alpha-actinin, which is a dimer, and then filamin here, which is also a dimer. And these help connect the actin filaments and give it some of its diverse functionality, right? You can have your sort of contractile bundle, your gel-like network, or your tight bundle. And we'll see sort of an example of that right here. So this is actually a contractile bundle in the sarcomeres of muscle cells, and you have alpha-actinin that basically separates the actin filaments in sort of a perfect size so that the myosin 2 motor can actually move here and allow for muscle contraction. So a motor protein can actually fit between these actin filaments. When you have a tight parallel bundle, that's actually cross-linked with fimbrin, and you can see that it's tight, and that actually won't allow a motor protein to enter into it. But these are actually what are sort of cross-linking for microvilli and filopodia, so that you have this tight packed bundle here. If you want to see a nice EM of alpha-actinin, so once again, this is what's cross-linking actin bundles in muscle cells. Here you can see it. It keeps them perfectly spaced apart, and half will bind to each. Okay, so this is just the EM of alpha-actinin. And we'll see alpha-actinin again when we cover the muscle cells in the sarcomere when we're looking at the motor proteins. But in the case with microvilli, what you end up having is the actin filaments packed pretty close together, and they're cross-linked with vilin and fimbrine here, and you can see, and then you have some other sort of lateral sidearm proteins like myosin 1 and calmodulin attached to the plasma membrane. And these sort of stick out in your intestinal epithelium to actually absorb nutrients, okay? And it helps just increase the surface area of the cells. And here's just a freeze fracture EM and staining of the microvilli. So here you can basically see the actin bundles in it, and they're all connected by the fimbrine that's down here. Okay, so it sort of holds it together. And then lastly, you have the filament dimers, which help cross-link the actin in the gel-like matrix, like the lamellipodia. Okay, so these will cross-link and cause the actins to form this gel-like matrix, which sort of allow the cell to sort of roll forward and push itself forward by the sort of addition of the subunits on the plus end by the formins and profilin. Okay, and if you have a defect in basically the filament dimer, you actually don't form the lamellipodia, so this is a melanoma cell line, and you can see they produce these little blebs here, but there's no lamellipodia. The second you take these cells and you sort of ectopically express the proper filament subunit that normally has a mutation in it, you can see that these cells actually form sort of nice lamellipodia, and they can move, and you can sort of see it right here. And then ultimately the actin is actually cross-linked to the plasma membrane, and one of the proteins that does that is this ERM protein, and so it's regulated by phosphorylation, so you can get phosphorylation of it, or you can get it binding to the PIP2, the phosphoinositol bisphosphate, and the plasma membrane, and it can also actually interact with transmembrane proteins like CD44, and you'll have your sort of membrane binding domain and alpha helix, and then your actin binding domain. So you'll get signals. If you go back to part A of this and look at the neutrophil chasing the bacteria, you know, all the signals are coming in for this to form and push and get the lamellipodia. And some of the ERM proteins are esrin, radaxin, and moesin here, and these help connect the actin to the plasma membrane. Okay, so that's pretty much all that I had for today. On Tuesday, we'll cover the motor proteins, and please stay up with the reading. And just so you guys know, the second exam is coming up on March 25th. This is the Thursday after spring break, and it'll cover lectures 9 through 15 for that. Okay, so I will see you all Tuesday, and I'll talk to you later. Bye-bye.